In our previous meeting, we discussed the paradigm shift in biomedicine. It used to be a hypothesis-driven enterprise, and it increasingly becomes a data-driven one. In this unit, we will discuss one main source of data, arguably the major source of data that enabled this revolution: the human genome. The human genome is over 3 billion nucleotides long. These nucleotides, the exact sequence of nucleotides, is what makes us human. The order of nucleotides is almost identical in all cells of all humans. If you check the sequence of nucleotides in 2 cells, taken from 2 random individuals, the sequence of nucleotides in these 2 cells will be more than 99% identical. Almost identical, right? In fact, it will be 99.5% identical on average. But this 0.5, this very tiny fraction of differences between any 2 cells taken from 2 random people, translate into a substantial number of differences. 2 people differ from one another in millions of points along the genome. A number of new technologies allow us to determine the sequence of nucleotides in cells, and sometimes even the exact sequence in an individual cell. This now allows us to identify all the genomic differences between 2 people, or even between 2 cells taken from 1 person. Sequencing machines around the world generate billions of sequences every day. Huge computers store these data. Within these sequences are probably answers to some of the most important and most pressing questions we have today. Why do some patients respond so well to certain treatments, for example, to cancer immunotherapy, while others do not? Why certain patients suffer from some diseases or conditions, while others, who may be exposed to the same environmental risks, do not? At least part of the answer to these questions is in the differences in the genome sequences. This is why so many doctors and scientists take samples from patients and send them to be sequenced. But sequencing machines reveal all the genomic differences, ones that affect our health and ones that may not. We don't always know which of these differences is consequential, that is, have a manifestation in the phenotype, and which are inert. We don't even know if there are differences that have no effect. How do these sequencing machines work? What kind of data they generate? And what can we do to separate the wheat from the chaff, to identify relevant differences and distinguish between them and the irrelevant ones? This will be one of the major topics of this course.